All right, so now we're going to start looking at the cell. So today we're going to just do kind of a tour of the cell. What are all the things that make up a cell? What different types of cells are there, right? What's the difference between an animal cell and a plant cell, and all that good stuff. So first we're just going to talk about just an introduction to the cell. What do all cells have in common? Who discovered the cell, the first cell? And then we're going to get into our two types, our broad categories of cells, which are our prokaryotic cells and our eukaryotic cells. So our pictures here, we have a prokaryotic cell over here, and then we have an animal cell and a plant cell. So these guys are both our eukaryotic cells. So who actually discovered the first cell? Well, it was Robert Hooke who was looking at cork, which is kind of like from an oak tree, like bark, and he was looking at it under the microscope and he saw these little kind of square-shaped little rooms, he called them. And if you think of little rooms, maybe in a jail, you would call it a cell, right? A small room. So that's how it got coined as a cell. And these guys are plant cells of an oak tree. So he was able to look under the microscope and just kind of was describing what he saw. And that was way back in 1665, so quite a long time ago. We've been looking at things under a microscope. And obviously back then the microscopes weren't as advanced as they are now. So nowadays, because we have so much better microscopes, we are able to really better understand the cell and the components of a cell. So there's a couple different ways we can actually look at things under a microscope. And the first and maybe most common way would be a light microscope. So you just shine a light through it. It's got to be a very thin slice of something to be able to see through it. And so our first picture here would be a light microscope picture of a paramecium here. And then the next kind, which you would see as much more magnified, would be a transmission electron microscope. So they actually bounce electrons off of it. And so you're able to see this transmission electron microscope. And if you notice, it's a lot more detailed on the stuff that's kind of inside the cell, right, compared to our light micrograph picture. And then last but not least, which has the highest magnification, and you don't need a cross section. So both of the light micrographs and the transmission electron micrographs need a slice, right? A cross section of tissue. The scanning electron microscope is actually looking at the surface. So you're taking the same paramecium over here and you notice there's kind of a fuzzy little border on it. And so we're looking at those fuzzy little hair cells coming off that paramecium, and we can actually really appreciate it in kind of three-dimensional, in a three-dimensional image, okay, at much higher magnification. So if you're interested, there's, a lot of classes that look under the microscope, and there is a whole plant biology class, and this is where a lot of these images come from. And these are all scanning electron microscope micrographs. So these are kind of cool if you want to look through them. Like those are some butterfly scales, a lot of pollen. You can look at that. They just look really cool, right? You don't think of things that would look so cool under the microscope. So now that we've, discovered what a cell is, and people started looking at a bunch of different living tissues, and what they discovered and came up with was this cell theory around, the mid 1800s. So what they observed was that all living things were made up of cells, and that cells are really the basic building blocks of life, right? And so the thing is that they thought that cells come from other cells. So a cell wouldn't just come out of nowhere, right? So, and that's, we're going to talk a lot more about division, cell division later in the course. So cells beget cells, right? And then, a lot of different branches of biology came out of the study of cells because they, started to realize that, hey, this is the basic unit of life. Then you get into cell biology, microbiology, even genetics and biotechnology. So now that we can see kind of the inner workings of everything, there's a lot more research that starts coming into play. So microscopes are great tools to help us see a whole range of cells and even structures that we wouldn't normally have been able to see. So we started discovering these smaller and smaller cells and even some molecules and compounds. So just to give you kind of an idea of the size range of things in the world, we have a size scale here on the left, and then we have kind of different objects in that size range. So things that we can see without a microscope or with the naked eye, we say, are things that we see every day, right? So humans are about, a little over a meter tall. Some even, so this is kind of cool, and you'll learn more about this if you take anatomy, but some cells in the body, like nerve cells and muscle cells, are super, super long. So especially nerve cells, they start in your brain or your spinal cord, and that little axon goes all the way out your limb, all the way to your fingers and toes, right? So long, long cells, almost as long as we are. And then you get into things like chicken eggs, frog eggs that are, we can see them, obviously. You could see it, maybe the frog egg with a microscope a little bit better. But, they're in the 1 centimeter to 10 centimeter range. Whereas the smallest cell that we can actually see with the naked eye is a human egg. So the ovaries in females produce the human eggs, and you can actually see it with the naked eye, and if you can believe it or not. Obviously, it's a lot better with a microscope, but you can observe it. So it's the smallest thing we can actually see with a naked eye. So it's the largest single cell in the human body. Whereas most animal and plant cells are in this range where we really need a microscope to appreciate, all the things inside the cell. And then we get into kind of some bacteria, even smaller bacteria and viruses where they may be even too small for light microscopes. And then you're getting into the electron microscope world. And we were talking a lot about these organic compounds last lecture, proteins and lipids. So these molecules, are going to need an electron microscope as well. And then you get into the world of teeny tiny, right? Things that we can't even see with an electron microscope. Things like atoms or small molecules like water, H2O. So just to give you an idea of the scale, of some of these size of cells. And we're going to mostly just be talking about plant and animal cells and their organelles. Okay, so in this range, 1 to 100 micrometers. So that little m is a micrometer. Okay. And then down here is nanometer size, so super, super tiny. So now let's talk about some features that all cells share. So we're going to talk about the differences between the cells as well, but what do they all have in common? And the first thing is a plasma membrane. So this is a boundary around the outside of the cell. Otherwise, all those organelles that are inside the cell would just fall out, right? So you have to have a containment of all those organelles inside the cell. So it's flexible though, so it can expand and contract, but it is important for the cell to keep everything inside. And the plasma membrane is made up of those phospholipids. So remember we talked about phospholipids last lecture as being one of our 3 different types of lipids. And the phospholipid, we said, had a hydrophilic head, meaning it likes water, and a hydrophobic tail. So what happens is most cells, many cells, live in a fluid environment, right? So that means that hydrophilic head is probably going to face outward to that fluid environment. But then we have that hydrophobic tail that does not like water. So what they do is they go back to back. So you have 2 phospholipids back to back, and they create what's called this phospholipid bilayer. And that phospholipid bilayer creates that plasma membrane. Okay, so if you just think of it, it continues all the way around in a circle around the cell or sphere, right? Or whatever shape your cell is, mostly spherical, right? And then what happens is there are these proteins and even cholesterol and sugars and things that are stuck inside that plasma membrane. And this is important for movement of things in and out of the cell, right? Communication, signaling, all kinds of things. And because that inner layer of that phospholipid bilayer is hydrophobic, meaning it doesn't like water, how do we get things that are hydrophilic across that membrane? And so these proteins can create channels and they can move things inside and out of the cell. But it definitely helps. It's kind of like a gated community, right? You let some things in and you keep other things out. So that's the whole job of these proteins in the plasma membrane. So another thing that all cells share, so we have a plasma membrane, right? We've got to hold everything in. And then there's got to be stuff inside the cell. And so the stuff inside the cell is called cytoplasm or cytosol, okay? And that's everything inside that plasma membrane, but not including the DNA. Okay, so the DNA is either housed in a nucleus or it has its own little area, and we'll talk about that in a minute. Okay, so the cytoplasm is everything. So including all the little organelles, everything except the DNA that's inside that nucleus, okay? Whereas cytosol, so they're somewhat interchangeable. A lot of people think they're the same thing, but cytosol is the actual kind of jelly-like fluid inside the cell. So cytoplasm is everything inside that plasma membrane, but the cytosol is just the fluid part in between all those organelles, okay? Now, all cells have DNA, and we call that chromosomes, right? So they all have chromosomes, they all have DNA. So they all have genetic material that is the blueprint to make all their proteins, right? So we talked a little bit about what DNA is, right? Our nucleic acids, one of our 2 nucleic acids, and it's that blueprint. So our cells read the DNA to create amino acids in certain sequences to create proteins, okay? They all have ribosomes. Okay, so what are ribosomes? Well, they are the structures that are going to be creating those proteins. Okay, so the chromosomes contain the DNA, which is the blueprints to make those proteins, and then the ribosomes are what are actually reading those blueprints to create those proteins. So the ribosomes, kind of think of them like the little factories. So they're going to be creating all those proteins. Okay. So let's pause the video for a little bit of review and answer the questions. So what are some of those common features that all cells have in common? So there's 4 of them, and see how many you can list. And then also, what is the cell theory? So hopefully you were able to pause the video and answer these questions. So what are those common features? Well, first, we have to have a plasma membrane. 2, we have to have stuff inside the cell, and that's called cytoplasm and cytosol. 3, we have to have some sort of DNA, and they're in chromosomes, right? And then 4, we have ribosomes, something to make those proteins out of the DNA. Okay, so those are our 4 common features that all cells have in common. And then what is cell theory? Well, cell theory is that all living things are made up of cells, and also that cells come from other cells. Okay? So now let's dive into our different types of cells, and the big broad categories are prokaryotic cells and eukaryotic cells. So let's talk about prokaryotic cells first. They're very simple. So they're structurally very simple, and they're very small. And the thing about prokaryotic cells and what distinguishes them from eukaryotic cells is that they lack a true nucleus. Okay, so if you break down the word prokaryote or prokaryon or prokaryotic, pro means before. Okay, so pro always means before, and a karyon is a nucleus. So this means before nucleus. Okay, they have DNA because we said all cells have to have DNA, they have to have chromosomes, so their DNA lives in an area called the nucleoid. Okay, so it's not a nucleus, it's a nucleoid. And it's just a cluster of chromosomes. Okay. And what the other thing is that yes, they have cytoplasm and cytosol inside their cell, inside that plasma membrane. Their organelles that are inside the cell don't have their own membranes. So they're organized in different regions, but they aren't delineated from each other like they are in eukaryotic cells. And you'll see the difference when we talk about organelles specifically. But prokaryotic cells lack membrane enclosed organelles. So they're just kind of in their own little areas of the cell. Okay. And what's unique to prokaryotic cells is that they have a cell wall. Okay, so they have a cell wall, and then inside the cell wall they have a plasma membrane. So as we said, all cells have a plasma membrane, but these guys also have a cell wall. Okay, so here's our cell wall, which kind of is the outer shell of the cell, and then inside that is that plasma membrane. And then inside that is all the cytoplasm, cytosol with all those non-membrane bound organelles. And here's that nucleoid, which is full of those chromosomes. And then the other thing unique to these guys are what's called a flagella. And it's like a little tail. So these guys have these little flagella that come off to help propel them through fluid. Okay? It's like a little tail, similar to sperm. Okay. So those were prokaryotic cells before nucleus, right? So they have a nucleoid instead of a nucleus. Now let's get into eukaryotic cells because they're a bit more complex And that's us, right? So we have eukaryotic cells, animals, and then also plants, okay? So these guys contain a nucleus, right? So they have nuclei that contain those chromosomes, that DNA. And we also contain organelles, membrane-bound organelles, okay? So this is a sample animal cell and a sample plant cell. They look very similar, but we'll talk about the differences. So let's go through some things that a classic animal eukaryotic cell has. So we've got our plasma membrane, right? So all cells have plasma membrane. All cells have cytoplasm and cytosol, okay? We have a nucleus containing the chromosomes inside. Okay, we have ribosomes. We said all cells have ribosomes, and essentially the ribosomes are those little tiny orange dots. Okay, so we find ribosomes kind of on the outside of the nucleus, on this weird blue stuff called endoplasmic reticulum. Okay, we also have free ribosomes just floating around in the cytosol. We also have these weird things called mitochondria. That's these big orange things, okay? And we'll talk about all the different organelles here in a minute as well, but just to give you a little tour, okay? And then compare that to our plant cell. The plant cell has a much more rigid structure. So it has that plasma membrane in here, but it's surrounded by a cell wall. Okay, so similar to that prokaryotic cell where that has a cell wall as well, a plant cell wall is a little bit different than a prokaryotic cell wall, but we have a cell wall. Then if you notice, okay, we've got a nucleus here, we've got cytoplasm, cytosol, but we've got this strange kind of clear structure in the middle here, and that's a vacuole, okay, full of fluid, full of water, and we'll talk about that. Then we've also got these green things. Okay, we do see a mitochondrion, which is in our animal cell, but now we also have these weird green things. Okay, those are called chloroplasts. Very important in plants and photosynthesis. So we'll talk all about photosynthesis in another lecture. Okay. So now let's talk about what we find in these eukaryotic cells. So we actually kind of group them into functional components. So we have 4 basic structures. And first we have a nucleus and a ribosome, okay? Because they kind of work together, right? The nucleus houses that DNA information for the ribosome to create those proteins. We have an endomembrane system, okay, and we'll talk all about that because those make some very important things. We have some sort of energy processing organelles. So we talked a little bit about mitochondrion and chloroplasts, so that's how the cells create energy. Okay, because think of a cell, right, like a little powerhouse. So it's creating proteins and other compounds, so it needs energy to support those processes. And then we have some sort of structural support, movement maybe, if the cell is able to move, and then also communication. How does it communicate with other cells in its environment? Okay. So first let's talk about the nucleus. So we have a nuclear envelope, which essentially encloses the nucleus, similar to what the plasma membrane does for the whole cell. We have a membrane-bound nucleus. So that's called the nuclear envelope. We have a nucleolus, okay, and that is the central region of the nucleus. So it's kind of this darker purple color, okay, inside the nucleus. And we also have chromatin. So chromatin is throughout the nucleus, and that's our DNA. Okay, so if we look at this whole picture, what are we looking at? Okay, so this is from the textbook, and we can see a transmission electron micrograph here of the surface of the nucleus. And it actually can show you the two different layers of the nuclear envelope. So we have an inner and outer membrane. Don't worry too much about that, okay? But that's what these two layers are of that nuclear envelope. And there are even little pores that span the two membranes to allow things to go in and out of the nucleus. Okay, and that's just what this blow up picture is, showing you those little pores, okay, in the nuclear envelope. Same with this. So this is just a close-up picture, even super close. And then we've blown up a picture of our chromatin. Okay, and we'll talk a little bit more about the chromatin that makes up our chromosomes and the DNA. Okay, but it's all spiraled in here in this unit called a chromatin. Okay. So let's look a little bit closer at that nuclear envelope. So we said it's double membraned, and we said it's similar to the plasma membrane. And what happens is it regulates kind of what is going in and out of the nucleus. So it doesn't want everything in there, obviously. So we have these pores that allow things to come in and out. The nucleolus is where we create ribosomes. Okay, so we create our own ribosomes that are then going to read all that DNA in the chromatin and create proteins. Okay, and the DNA is in those chromatins. Okay, so the chromatins then create those chromosomes, and we'll talk a little bit more about that. So now let's talk about ribosomes. Okay, so these guys, we said, think of them like a little machine or a little powerhouse that builds the proteins for the cells. So we have ribosomes that are free, which are suspended in the cytosol, right? So they might be out here in amongst all the other organelles. But then we also have ribosomes that are attached to either the endoplasmic reticulum, which is that blue stuff, or even that nuclear envelope itself around the nucleus. So those are the bound ribosomes. So if we look at a transmission electron micrograph here, so we've blown up an edge of that endoplasmic reticulum, we can actually see the little dots of ribosomes. Okay, so these guys, some of them are free, right? And then some are actually stuck to this endoplasmic reticulum. So the lines are the endoplasmic reticulum. And then the ones that are stuck on there are the bound ribosomes, and then the ones hanging out in the middle are the free ribosomes. And this is just a drawing of what a ribosome looks like. Okay, and then here's a kind of a computer rendition or model of it, because we said, you know, it's just a large protein, but it also produces proteins. So it reads our DNA to produce proteins. Okay, so it's a very large molecule that creates proteins for our body. So what is the endomembrane system? So we have a picture here. Here's our nucleus with that nuclear envelope, and it actually is a continuation of the nuclear envelope here, and that's our endoplasmic reticulum. And we have those ribosomes stuck on there too. Okay, so we think of this with the Golgi body, which is over here, as kind of our warehouse shipping center. So once we produce all these materials, we're going to ship them either to different areas in the cell or even outside the cell. Okay, so it's kind of our production, shipping, delivery system of the cell. So let's look more closely at that. So first, the endoplasmic reticulum has two different sections. We said it's continuous with the nuclear envelope, but we have what's called the smooth endoplasmic reticulum, meaning it doesn't have any ribosomes attached to it. So if we look down here at our picture, we have another transmission electron micrograph, and here's the smooth ER, okay, and then here's the rough ER. So rough endoplasmic reticulum has those little studded ribosomes on it, creating kind of a roughened surface. You can think of it like that. And they have two different jobs. So they're continuous with each other and with the envelope, but they have different jobs. They produce different things. So the smooth ER, what it does is it synthesizes lipids. So what are those lipids we talked about, right? Our fats, our phospholipids, our steroids, all those guys, okay, are synthesized by the smooth endoplasmic reticulum. But they also metabolize carbohydrates and detoxify drugs and poisons. So that's kind of interesting as well, okay? So you might find these in the liver, you know, cells with very well-formed smooth endoplasmic reticulum. It does store some calcium ions. Most of our calcium is stored in our bones, but we can store some calcium in the smooth ER, okay? But really think, you know, producing lipids, okay, producing those fats. Now the rough ER has the ribosomes, okay? So what happens is the ribosomes are creating all the proteins, but what is the actual rough endoplasmic reticulum making? Well, it secretes glycoproteins. So if you break down glycoprotein, what is a glycoprotein? Well, we know now kind of what proteins are, and glyco, think glycogen, right? Maybe sugar, glucose. So it's a sugar protein, okay? And what it does is it distributes these little transport vesicles. So any of the substances made in the ER are going to go into these little vesicles. And then it's going to go out to other parts of the cell or out to the outside the cell. It can go three different ways, and we'll talk about it. And one of the ways is to the membrane. So it's helping to make and maintain the plasma membrane of the cell, okay? So the last part of this endomembrane system is the Golgi apparatus. I know that's kind of a weird word, right? The Golgi body or Golgi apparatus. And so what the Golgi body does, or Golgi apparatus, is once everything is being made and ships out from the endoplasmic reticulum, the Golgi body receives everything, okay? And it either changes it, sorts it out, and ships it to a certain destination of where it's supposed to go. So is it supposed to go inside the cell? Is it supposed to go to the plasma membrane? Or is it supposed to go outside the cell? So now let's talk about lysosomes. So we think of lysosomes kind of like the digestive system of the cell. So obviously it's not the same as our digestive system, like a stomach and intestines. Essentially what it is, it's a sac, a membranous sac of enzymes. Okay, and the enzymes are what are going to break things down. So we said enzymes are proteins, right, that like to break things down or even build things, but in this case, they break things down. And so what happens is we have our little lysosome here full of those digestive enzymes, and maybe the cell brings in some food from the outside, creating this food vacuole. So vacuole is just kind of a movement vesicle, right, to carry things around. And it's part of the plasma membrane, and we'll talk a little bit about that in a minute. And what happens is it joins with that lysosome, and then the lysosome, those digestive enzymes, break down the food so that you're able to, the cell can use those components from the food for energy. Okay, so not only does it digest food, but it also can break down damaged organelles. So say you have, a mitochondria that's damaged, it can join with a lysosome and break it down. Okay. So, your cell has to be able to produce things, but also break things down and keep things running smoothly. You don't want any sort of damaged organelles in the cell. You want to be able to get rid of them and replace them. So I have a really cool video here. Either you can copy and paste the link, or I will post the link for the video on Canvas as well. It's kind of cool to see how our cells create these food vacuoles by a process called phagocytosis. Okay, and that's this creating a little vacuole with the plasma membrane full of food. Okay, it's how our cells eat things, if you think of like quote unquote eating something, and that's called phagocytosis. Now, when we talk about eukaryotic cells, we also have what's called vacuoles. So we already talked about food vacuoles. So our cells can eat something and create a food vacuole using that plasma membrane. That whole process is called phagocytosis. But we also have contractile vacuoles. And essentially these are found in freshwater protists. And protists are just these single cell organisms that live in the water. And so they're able to hold water, and then they can squeeze the water out, or contract them, right? And kind of propel themselves using water. And the other kind is a central vacuole. And these guys are the vacuoles found in mature plant cells. So remember we said in the plant cell we had that kind of strange clear structure that holds fluid. Well, that's a central vacuole. And it's going to hold organic compounds, but mostly think of it like just holding water, like a water bladder in the cell. Okay. Now, another very important organelle found in both plant and animal cells, but it's a eukaryotic organelle, is mitochondria. Okay, so we find them in both. And essentially, this is the site of cellular respiration. And what is cellular respiration? Well, you've heard the term respiration before, right? We all are respiring or breathing, right? Bringing in oxygen, expiring carbon dioxide. But this is something a little bit different. Okay, so cells, this is how we turn food into chemical energy in a cell. So remember we said this is like the little powerhouse of the cell. Okay, so if we look, here's what a mitochondria looks like. It's those little orange guys. And they almost look like their own little cell. Okay, they have kind of a plasma membrane, an inner and outer membrane. They have free ribosomes inside the cell, or inside the ribosome. So it's kind of cool that they are almost like little cells, but they live inside our plant and animal cells, creating energy for us in the form of ATP. And we will be a lot more familiar with cellular respiration and ATP in another lecture. So now let's talk about that weird green structure that we find in plant cells and algae. And these guys are the site of photosynthesis in plant cells. So whereas mitochondria is able to convert food into energy as ATP, these guys are converting solar energy into chemical energy, into ATP. And that chemical energy is able to feed the plant. Okay, even though plants also have mitochondria as well, so they can do both cellular respiration and photosynthesis. And the cool thing about chlorophyll is that it is green. So it actually gives plants the green color. Okay, so it's a kind of a similar structure like a ribosome. It has an inner and outer membrane, has these little stacks of these green thylakoids is what they're called. Okay, they even have those little ribosomes inside them. Okay, and then that's an actual transmission electron micrograph of a chloroplast, which is kind of cool. Okay, so these are only found in plant cells, not in animal cells. So we've been saying, well, the mitochondria and also these chloroplasts look kind of like little cells themselves, right? But they live inside of our eukaryotic cells. So there's a theory called the Endosymbiont Theory, and it says that probably early ancestors of eukaryotes actually engulfed bacteria and kind of ate bacteria, and some of these bacteria actually stayed inside those eukaryotic cells. And it was this symbiotic relationship where the eukaryotic cell provided protection for that bacteria, and then the bacteria produced energy for the cell. So they kind of came to a agreement and said, hey, if you let me live in your house, I'll cook you food, right? So it's kind of this interesting theory that these, ancestral eukaryotic cells actually, ate this bacteria, which then the bacteria evolved into mitochondria and chloroplasts, which is really kind of a very cool idea, I think. But we see symbiotic relationships all the time in the world in biology. So we'll see a lot more of them in this class as well. We'll talk a lot more about symbiotic relationships. Right? So what are some arguments for this theory? Is that, well, they kind of look like bacteria, right? So they have their own DNA. They have their own ribosomes. They can grow and reproduce independently from the cell. So they're their own little cell, essentially. And they even have double membraned, right? We said that plasma membrane has an inner and outer membrane. And they're about the same size as bacteria. So really, this theory is very strong, and it probably is what happened and how we created mitochondria and chloroplasts. So eukaryotic cells also have what's called a cytoskeleton. So essentially, we've seen the word skeleton before, right? We have a skeleton made up of bones, but theirs is made up of different types of fibers and filaments. But essentially, it does the same thing. It gives the cell structure. And it also allows for different movement and activities within the cell in kind of a framework or a network of fibers. So cyto, just cyto, anything with cyto is referring to a cell. So it's the cellular skeleton. Okay, and so these are just our three different types of cytoskeleton fibers, and you don't need to know the difference of these or anything like that. Just know what a cytoskeleton is. So we have these microtubules, and so this is the cell itself. So you can see that it maintains that structure of the cell. It gives the cell its structure. We have microfilaments, and these guys kind of allow for movement within the cell. And then we have intermediate filaments as well. So these guys are all different fiber networks inside the cell. Okay. So what does the cytoskeleton do? So besides creating structure for the cell, it also allows for movement. So we talked about flagella on prokaryotic cells, but we also find flagella and what's called cilia on our eukaryotic cells. And they are these microtubules that are actually projections from the plasma membrane. So they project out from the plasma membrane, but they're made up of the cytoskeleton. And so an example on a eukaryotic cell is the flagella on a sperm cell. Okay, so that allows for swimming. Okay. But we also find what's called cilia, and you can think of cilia as like these long hair fibers. And essentially they have their own little movement. And so they make this little power stroke movement, and they are able to push things in a certain direction. Okay, so they move mucus and all kinds of things around. So we have cells in our body that have cilia to move mucus around. Okay. So if we compare cilia and flagella, cilia is a lot shorter, it's more hair-like. And so here's two scanning electron micrographs, one of the epithelium, the epithelial tissue in our trachea, and it has the cilia on there. So all these little hair fibers are cilia, and essentially they're going to move, they're going to have the mucus trap any sort of air particles and then move that mucus through the trachea and out of the respiratory system. And then the flagella, so here's a sperm cell here trying to penetrate an egg, and it has that long flagellum. So it's a lot longer, and there are a lot fewer of flagella in the world. Okay. So now when we're talking just specifically about plant cells, we see the cell wall. Okay, and it's an extracellular structure, and what that means is it's outside the cell. So we said the cell has a plasma membrane, and that's really the outer layer of the cell. But then we've added this extracellular structure called a cell wall in plant cells. And it is composed of cellulose mostly. So remember we talked about cellulose as being one of our polysaccharides or our carbohydrates. Okay, so polysaccharides, as we were saying in our other lecture, is a carb, right? Carbohydrate. So it's just a chain of glucose molecules, but it has a very strong, tough structure. So it's used as a protective structure in the cell wall of plant cells. Okay. And we can see that central vacuole pretty well inside the plant cell as well, and those little chloroplasts. Okay. So it's just this outer layer around the outside of the cell. Okay. And then that cell membrane inside, which is that kind of orange line. Okay. So finally, let's do a bit of review. Okay. So we talked about a lot of similarities, but also some differences between prokaryotes and eukaryotic cells. Okay. And we said, what are those things that are in common in all cells? And then what are the differences between those prokaryotes and eukaryotes? And then also what are some similarities and differences between animal cells and plant cells? So go ahead and pause the video, give yourself some time to answer. All right, so hopefully you paused the video and jotted down a couple of similarities and differences. So first, if we were talking about a prokaryote versus a eukaryote, so some similarities are the plasma membrane, right? So we have a plasma membrane. It's essentially those four similarities between all cells. So we have a plasma membrane, we've got cytosol and cytoplasm inside the cell, we have some DNA, right? And we have ribosomes, okay, creating proteins. But we also saw, okay, maybe some flagella, some cilia, that maybe aren't on all eukaryotic cells, but that could be a similarity as well. We have a cell wall on a plant cell. We said prokaryotes have a cell wall as well. So there's some similarities. But what are the major differences? Well, it has to do with the nucleus, right? So eukaryotic cells have a nucleus. The prokaryotic cell does not. It has a nucleoid. So just an area where the chromosomes sit. And then all the organelles are also membrane bound. So they're delineated from the other organelles in the cytosol. Whereas in the prokaryote, they're just kind of free floating around in different regions of the cell, but don't have plasma membranes. Okay. Now let's look at our plant and animal cells. What are some differences? So we know kind of the similarities, right? So they both got a nucleus. They've both got an endomembrane system, that Endoplasmic Reticulum and Golgi Apparatus. They both have mitochondria. They both have a cytoskeleton. Okay. Now, what are some differences? Well, the plant cell has chloroplasts and a central vacuole, as well as that cell wall. Okay, so plant cells are a little bit different than our animal cells. Okay. All right, and I think that's it. So if you have any questions, let me know. Otherwise, hopefully I'll see you guys in office hours. Thanks so much.